Hello, everyone. I'm Dina, and thank you for joining us today for the Illumina RNA Prep with Enrichment Introduction webinar. In today's webinar, we'll go over these 5 topics. First, we'll cover the advantages of Illumina RNA Prep with Enrichment, then talk about the specifics of the library prep and enrichment workflows, touch on the sequencing and analysis, and finally go over reagent kit configurations. So let's get started with the advantages of Illumina RNA Prep with Enrichment. RNA sequencing is an extremely powerful tool for understanding the transcriptome and is by far the most cited NGS method. RNA-seq can be used for gene expression profiling, transcriptome discovery, and transcription detection, making it an accurate, sensitive, and reproducible NGS method. RNA-seq is used in many different areas of disease research, including oncology, genetic disease research, and infectious disease research. An example of an infectious disease application would be using a respiratory viral enrichment panel with RNA extracted from wastewater samples to monitor infectious diseases such as SARS-CoV-2. And we'll talk in more detail about different enrichment panels and their content. Illumina's newest portfolio of RNA sequencing kits includes Illumina Stranded Total RNA and Illumina Stranded mRNA and Illumina RNA Prep with Enrichment. The Total RNA and mRNA-seq kits are going to give a very wide view of the transcriptome. In contrast to the targeted enrichment of Illumina RNA Prep with Enrichment, which allows us to narrow our focus to only the genes or regions we're interested in. For example, we can focus on the coding RNA exome or cancer-related genes with the enrichment workflow. So now let's talk about some of the advantages. This slide summarizes some of the key features for the Illumina RNA Prep with Enrichment kit. One of the great benefits when compared to legacy enrichment kits is that this is a fast workflow. This is due to both the tagmentation-based library prep and a single enrichment reaction. For experienced users, this will be about a 9-hour workflow with about 2 hours of hands-on time. The library prep is also automation friendly, so this is another way you can save time in the lab. If you're interested, I recommend visiting Illumina's automation website linked here for a list of the compatible platforms. The workflow is compatible with many different sample types, including high-quality RNA and FFPE or otherwise degraded RNA, down to DV200 values of 36.5. The library prep utilizes Illumina's unique dual indexes, with 192 currently validated, and 384 will be available in the near future. This is great for users sequencing on a high output platform like the NovaSeq 6000 and NextSeq 2000. And then we have flexible panel options. Currently validated panels are the Illumina Exome panel, viral panels including the respiratory virus oligo panel V2 and respiratory pathogen ID AMR panels, and then we could also use custom or third-party panels. Finally, Illumina RNA Prep with Enrichment provides a complete workflow from sample prep to analysis. And we have several analysis options available, including the DRAGENT RNA pipeline, DRAGENT differential expression, and DRAGENT pathogen detection apps, which we'll talk more about in the sequencing and analysis section. So let's take a brief look at the workflow now. Here we have an illustrated look at the full library prep workflow, starting with the pre-enrichment library prep on the left, followed by the enrichment workflow on the right. The pre-enrichment workflow features tagmentation of the cDNA with eBLTL beads rather than fragmentation of RNA before cDNA synthesis. For the enrichment workflow, we then pool the libraries before going into enrichment and capture of our regions of interest. So here are the validated panels. Illumina has tested and validated the Illumina Exome panel, which includes comprehensive coverage of coding RNA sequences, the respiratory virus oligo panel V2 or RVOP V2, which includes detection and characterization of common respiratory viruses, including coronaviruses and influenza viruses, and the respiratory pathogen ID AMR enrichment panel bundle, which is RPIP, which targets more than 280 respiratory pathogens, bacteria, and fungi, in addition to more than 1,200 antimicrobial resistance alleles. So note that both the RVOP V2 or RPIP panels can be used for sequencing the SARS-CoV-2 genome, as well as recent flu strains. And for a full list of what's included in the panels, check the product links in the resource slides that I provided at the end. Illumina RNA Prep with Enrichment can also be used with custom and third-party panels. Illumina does not have specifications for use with custom or third-party panels, so just modifications may be required. If you're interested in a custom panel option, please reach out to your local sales team or contact us in tech support, and we'll put you in touch with your local sales team. For any users who may have previously or are currently using the TruSeq RNA Exome Kit, I wanted to highlight some of the advantages of the new Illumina RNA Prep with Enrichment Kit. So the assay turnaround time for TruSeq RNA Exome is 2 days, whereas the Illumina RNA Prep with Enrichment workflow can be completed in about 9 hours. TruSeq library prep chemistry always involves full-length adapter ligation, but the new kit utilizes tagmentation to add partial adapters, and then the index PCR completes the adapter. Again, the indexes for the Illumina RNA Prep with Enrichment Kit are unique dual indexes, up to 192 currently, whereas TruSeq RNA Exome is only validated for single indexes, which are limited to 24 total. With the new kit, we move to 1 hybridization reaction versus 2. And a note on the reagent, the reverse transcriptase enzyme is included in the new library prep kit, whereas it is a user-supplied consumable with the TruSeq RNA Exome kit. So in summary, the new kit is 50% faster and 80% less hands-on time due to these workflow changes. So let's get into the details of the library prep workflow, or the pre-enrichment library prep. Here again is our illustrated look at the full library prep workflow, and we'll be talking about each step of that pre-enrichment library prep in detail on the left. This is the workflow diagram from the reference guide, which is a nice step-by-step breakdown of how we're constructing the libraries. So the pre-enrichment workflow has 3 main steps. First, we have to reverse transcribe the RNA to double-stranded cDNA. Next, cDNA is tagmented and amplified to complete the index adapters, and then the libraries are normalized. So step 1 is cDNA synthesis. This requires 10 to 100 nanograms of input. If we're using RNA extracted from FFPE or other degraded RNA, we need a minimum of 20 nanograms to start. And please QC the RNA input to ensure it has DV200 values of 36.5 or greater. For exome enrichment, we will want to start with purified total RNA. But if we're using the RVOP-V2 or RPIP panels, we have the option of starting with total nucleic acid or total RNA. We want to be sure we quantify the RNA input using a fluorometric method specific to RNA, such as qubit. And for exome enrichment, we recommend a DNase treatment on the input, and this is typically done during the RNA extraction process. If we're using a viral panel, since these panels contain both DNA and RNA targets, we do not want to include that DNA step if we're using total nucleic acid here. The table on the right lists some RNA handling best practices. We want to avoid multiple freeze thaws of that RNA input, and then avoid extended pauses until your RNA is converted to double-stranded DNA. We want to keep the thawed RNA and reagents on ice while we're doing the library prep, and then ensure the thermocycler lid is preheated for each synthesis step. Above the table here, we see the steps from the workflow diagram. And note that completion of CDNA synthesis is a safe stopping point. So if you'd like to proceed with the library prep the next day, you can check the reference guide for those storage details. So next, we have the Tagment and Amplify CDNA step. This step uses Enrichment bead-link transposome large, or EBLTL, to simultaneously cut and tag the cDNA before we amplify with index PCR primers to complete the library. Note the link on the bottom left. This is a bulletin that explains the different bead types in our Illumina library prep kits. The EBLTL is specific to the Illumina RNA Prep with Enrichment kit. So if you use different Illumina kits in your lab, make sure we're grabbing the correct bead type here. And let's review some best practices and tips in the table. Bead handling best practices to keep in mind. Always bring the beads to room temperature before use and fully resuspend. Prepare fresh 80% ethanol for the wash step. And do not allow the beads to fully dry, as this can lead to low library yield. We also want to ensure that these beads do not freeze during storage, and they're stored at 4 degrees C. The index adapters plate, so these indexes are pre-mixed in single-use wells, and this is to reduce the risk of cross-contamination. And then the number of PCR cycles here does depend on the RNA input quality or the panel used, with FFPE, degraded RNA, and the RVOP panel needing 17 PCR cycles. In the workflow diagram on the top right, we do have 2 safe stopping points after each step here. And again, we can refer to the reference guide for details on storage temperature and how long the DNA can be stored after each step. So the indexes used in this library prep are the IDT for Illumina, DNA, RNA, UD, or unique dual indexes. They're a little different than some of our other index kits in a few ways. The I7 and I5 indexes are both 10 base pairs, and they're pre-mixed within each well. And so each well is a single use to reduce the risk of cross-contamination. Like with any library prep, if we're pooling a low number of samples for sequencing, we need to consider the color balance of the indexes on the instrument. With these indexes as a pooling strategy, we recommend pooling libraries down a column for ideal index color balance. For example, the first 2 or last 2 wells in a column. And this guidance can be found in the index adapters pooling guide linked here. I also recommend checking out the bulletin linked at the bottom if you're interested in understanding more about the unique dual indexes. The final step of the library prep is to normalize, which prepares the libraries for enrichment. We do need to quantify the libraries using qubit double-stranded DNA broad range. You can also QC to check the library size at this point, but this is optional. The expected average library size is about 380 to 450 base pairs. After quantifying each library, we can now pool for enrichment. So we have the option of 1-plex, which is 1 library for 1 enrichment reaction, or 3-plex, which is combining 3 libraries for a single enrichment reaction. And there's a link to a technical bulletin here that goes over understanding enrichment plexity. So for 1-plex, we want 200 nanograms of the library diluted to a volume of 7.5 microliters. For 3-plex, we want 200 nanograms of each library diluted to a volume of 2.5 microliters for a total of 7.5 microliters of libraries. The table here is taken from the reference guide, and this just highlights that the mass is 3 times as much for the 3-plex enrichment reaction. However, the volume is the same for either plexity. And if your total volume is greater than 7.5 microliters at this point, we can concentrate by vacuum or filter. So now we're ready to take our libraries into the enrichment workflow. So the first step is to hybridize the probes. This is where we take the complete library that we normalized or pooled and add the biotinylated probe panel to bind our regions of interest. Again, our probe panel options include the Illumina Exome panel, RVO-PV2, RPIP, and then customer third-party options. Some best practices to keep in mind for this step include preheating your microheating system to 50 degrees C. This is to heat that NHB2 buffer. And we want to ensure correct handling of the NHB2 buffer, including preheating it for 5 minutes, fully resuspending it, and then keeping it warm to prevent any precipitate formation. And then we'll always add our hybridization reagents in the order listed in the reference guide. And for hybridization time, we do have flexibility here. We can go for 90 minutes or up to 24 hours if you want to set that reaction up and let it go overnight in the lab. That's a possibility. After we hybridize the probes, we need to capture the probes. So we're going to wash and wash and wash and wash and elute. Some of the best practices to keep in mind at this step, we want to ensure we're using the correct beads, which will be SMB3 or streptavidin magnetic beads. We'll use the bead handling practices we went over already, and we will need to preheat the microheating system again for the EEW reagent, this time to 58C. Note that the thermocycler will be running continuously for these capture and wash steps. We want to prepare fresh elution master mix with HP3. And finally, the transfer wash here is critical. So do not skip the plate transfer step for the final wash, as this can negatively impact our library yield. Once the libraries are captured, this is a safe stopping point. After the capture, we are ready to amplify and clean up the final library. We can perform the final library QC to check the enriched libraries, and we do expect a final library size of approximately 380 to 450 base pairs, and that corresponds to a 200 to 250 base pair insert size. We also need to check the concentration of the final libraries using a fluorometric method such as qubit, and we expect at least 1 nanomolar library yield. So that completes the library workflow. So I hope the breakdown of these steps was useful. So let's talk about sequencing and analysis options now. The general steps in the sequencing and analysis workflow that we'll touch on are the sequencing that we'll touch on for Illumina RNA Prep with Enrichment are the sequencing recommendations, the instrument run setup options, and finally, data analysis considerations. So here are the platform-specific and panel-specific sequencing recommendations. If we're using the Exome panel, our read length will be 2 by 100 base pairs. And since this is a large panel, it is better suited for higher throughput platforms like the NextSeq 550, NextSeq 2000, and NovaSeq 6000. Note that the maximum number of samples per run here is based on 25 million reads per sample. If we're using the RVOP V2 panel, our read length will be 2 by 75 base pairs. This panel is smaller, and we're targeting just 1 million reads per sample, so this is a great option for the MiSeq, MiniSeq, and NextSeq 550 again. You can check the sequencing coverage calculator linked at the bottom if we want to run on a platform that's not shown in these tables, or if you have different coverage requirements for your application. For a quick overview on how to use the sequencing coverage calculator, check out the support short video linked here as well. So let's talk about loading concentration briefly. This table is taken from the reference guide. And note that the MiSeq and MiniSeq recommendations are specific to the RPIP panel. All of these loading concentrations are starting recommendations, so they may require further optimization in your lab. And we have a support short that goes over how to choose a loading concentration if the platform you are planning to use is not listed here in the table. And for directions on how to prepare the library pool for sequencing, please see the platform-specific system guide or denature and dilute libraries guide. Here we have links to different run setup options depending on the platform you're using. The Illumina Experiment Manager software can be used to make a sample sheet for various platforms, and I included a link to the most up-to-date version here that will have Illumina RNA Prep with Enrichment included in those drop-down menus. NextSeq 1000 and 2000 users can refer to the instrument run setup link. And for platforms that are running local run manager, there's a link with more details about setting up a run in LRM. So we are now ready to sequence and generate data for analysis. So there are several analysis options depending on the panel we're using. For any sequencing project, the first step is to generate FASTQs, which is part of the sequencing platform workflow. So your FASTQs will be used as input for all of these analysis options. For the Exome panel, we can use the DRAGENT RNA pipeline and DRAGENT differential expression apps. We also have RNA-seq alignment and differential expression apps available in BaseSpace. For RVOP V2 panel users, we can use the DRAGENT RNA pathogen detection app or the ID by DNA Amplify RVOP platform. And for the RPIP panel, we have the ID by DNA Amplify analysis software. So I wanted to highlight the DRAGENT RNA pathogen detection app with a link at the top here if you want more details. This app uses a combined human and virus reference to analyze pathogen data. It can be used for libraries enriched with that RVOP panel. And on the right here, we have an example sample report showing the detection of SARS-CoV-2. So this is a nice option for your viral genome sequencing. And if you want an in-depth look at the DRAGENT RNA pathogen detection app, you can see our webinar Metagenomics Part 2 Introduction to Analysis linked here. They go over this in detail there. Here is the DRAGENT RNA pipeline, which is what we want to use if we've enriched with the Exome panel. The DRAGENT RNA app performs secondary analysis of RNA transcripts and offers multiple operating modes, including reference-only alignment and annotation-assisted alignment with gene fusion detection. And there's a link at the top if you'd like more details about the app. And then if we run our sample through the DRAGENT RNA app, we then have the DRAGENT Differential Expression app. So first the data goes through the DRAGENT RNA pipeline, and then those output files can be used as input for differential expression analysis with this app. If you want to take a look at some example data, we have demo data available in BaseSpace. Note that these demo data sets are free to import and do not count against your storage limits. To navigate to the demo data, click the BaseSpace link at the top of the slide and log in. And then click the Demo Data tab on the right. And use the search bar to type in Illumina RNA Prep with Enrichment. This will bring up both Exome and RVOP data sets available from various sequencing platforms. If you're interested in that RPIP panel specifically, the second link at the top here will take you directly to that demo data and demo data report from the ID by DNA Amplify app. Now let's talk about the library prep kit configurations and what needs to be purchased. This slide lists the required reagents. So we need to pick one item from each box shown here for the complete library prep. First, we have the library prep kits that come in 16 or 96 sample kit configurations. And take note of those blue arrows. The 16 sample kit was designed for 1-plex enrichment reactions, or one library for one enrichment reaction. The 96 sample kit was designed for 3-plex enrichment, or 3 libraries pooled together for one enrichment reaction. So that 96 sample kit contains enough enrichment reagents for 32 enrichment reactions. Next, we have the indexes. Currently, there are 2 sets of unique dual indexes available, with 2 more expected in the near future. And last, we need to purchase the enrichment panel. I included the number of reactions that each enrichment panel tube is good for here. So the Illumina Exome panel is good for 8 enrichment reactions, and the RVOP V2 panel is good for 32 enrichment reactions. If you need more library reagents or enrichment reagents for your experimental needs, we can purchase either additional pre-enrichment library prep reagents or the RNA fast hybrid reagents, both of which are available in 16 sample configurations. So here's the list of user-supplied equipment, and this full list can be found at the end of the reference guide linked here. The list is split to show the equipment that's required in the pre and post PCR areas for your workflow. And most of the equipment is probably familiar to you, like thermocyclers. You can check that list for the validated models that we tested, 96 well PCR plates, magnetic stands. But I wanted to highlight the microplate shaker that is required. It should be capable of reaching 2,700 RPM, and this is critical as it's used in various bead steps throughout the workflow. So now let's go over our kit resources. The links at the top are for the RNA Prep with Enrichment product page, where you'll find the library prep kit reagents and catalog numbers for ordering. The second link is for the product support page, which I'm showing a screenshot of here. The tabs at the top of the support page are used to navigate our different resources. So let's take a look at what we can find in some of these tabs. The documentation tab contains that downloadable reference guide, as well as app notes and a product data sheet. If we scroll down that documentation page, you'll find a section called Product Literature. And here there's a great app note about using the RVOP-V2 panel for SARS-CoV-2 detection and characterization. And there's another great app note a little further down the list, not shown in the screenshot, about wastewater surveillance for SARS-CoV-2. The Contents and Storage tab lists the boxes and reagents in each library prep kit, plus those storage temperatures. And finally, there's a training tab that has links to relevant recorded webinars and a 30-minute training course that covers pretty much the same material we covered here today, just in a different format. We also have technical bulletins on the Illumina support page, and I listed the bulletins that were linked throughout the presentation today, as well as one on DNA and RNA isolation considerations for Illumina library prep kits. Next, we have the training support page. So on the screenshot here on the left side of the web page, you'll find the training type menu. And if you click to expand that menu, you can see the different training resources available, so you can narrow down what you're interested in more easily. And I wanted to highlight the support shorts, some of which I linked throughout the presentation today. These are short 2 to 5 minute videos that cover various topics in library prep, data analysis, and sequencing platforms. And then those video troubleshooting tips, those are short videos for the different sequencing platforms that you may find useful. And of course, we have pre-recorded support webinars linked on the training page as well. So thank you so much for taking the time to join me today. I hope this session has been helpful for you.